So Marc Torra works at the UG group of clinics. He got his degree in biology at the University of Barcelona, then went on for a master's in cytogenetics and reproductive biology at the Autonomous University of Barcelona, and he and then he got his PhD corrected to a correction between UV and Eugene, and actually one of his supervisors who was Rafael Oliva. So being the theme of these symposium, fathers and sons, he have an academic father and son pair somehow, and Marc is going to sort of widen the focus on the previous presentation and cover other aspects of the genetics of male-related fertilization failure. We can see perfectly your presentation, Marc. So the floor is yours.
Okay, thank you for the introduction. Today I will talk about the genetic alterations that can explain fertilization failure after ICSI in the male. Before all, if we want to talk about fertilization failure, we really need to know what fertilization is. By definition, fertilization is the biological process by which the male and the female gametes, the sperm and the oocyte, fuse together to form an embryo, which is able to start its development. Especially thanks to in vitro fertilization, we know that fertilization is not complete when a sperm binds to the oocyte. It's not complete when the sperm penetrates inside, but fertilization is complete once we see a clear formation of two pronuclei. And what do we need, or what does the sperm need for successful fertilization? Well, apart from being able to move, there are some structures which are essential for fertilization. For example, the acrosome, the centrosome, which is essential for the zygotic events. And also there is a region of a special importance, which is the perinuclear theca. This fraction of the sperm is located between the acrosome and the nuclear envelope, and it's important for the activation of the oocyte because it contains oocyte activation factors such as PLC-zeta. In the context of infertility, in a fertility clinic, this oocyte activation, this fertilization happens in the context of IVF, for example, by using ICSI. As you know, ICSI is one of the main techniques in many countries, including Spain, and is very efficient in terms of fertilization rate, which can reach means of 75%. However, in some times there is fertilization failure. This fertilization failure is usually characterized by lack of oocyte activation failure. But the day after ICSI, we can also observe the presence of zygotes with 1PN or 3PN, something commonly referred as abnormal fertilization. We know that there are some factors which are associated with a higher incidence of fertilization failure after ICSI. For example, when we inseminate a low number of oocytes, when we have a previous existence of cycles with fertilization failure, when there is a male factor, for example, severe astenosospermia or the use of testicular sperm, when we inseminate in vitro matured oocytes, which have less ability to be activated, or although not fully confirmed, we tend to blame always the oocyte and the advanced maternal age. But we also know that fertilization failure also happens in absence of these factors. For example, when we have normosospermia or we inseminate donor oocytes with proven fertility. And the truth is that for many cases of fertilization failure, we don't really know which is the origin. This implies not only a difficult clinical management, but of course, a complete lack of chances to become pregnant, which cause psychological impact and economic impact for the patients and higher chance of treatment discontinuation. The problem is that right now there are very few methods to to see the, sorry, I lost the presentation. Let me check. Can you see the presentation now?
Yes.
Okay, thank you. So as I said, the problem is that there are very few methods to diagnose fertilization failure. One of them, for example, is the use of MOCA or HOCA. This method uses mouse or human oocytes to evaluate the sperm ability to trigger oocyte activation by analyzing the calcium patterns generated in the oocyte. However, this technology is not always available in many laboratories because it uses a biological material which is not easy to obtain, which are the mouse and human oocytes, and also it requires specific equipment and protocols. So for this reason, today I will talk about gene sequencing, which is an approach which is starting to be applied for the diagnosis of infertility as well as fertilization failure in particular. The advantages of of gene sequencing is that it's fast, it's relatively cheap, can be provided by an external service, so you don't need specific equipment in your lab. You only need a small amount of biological material, even single cell. You don't need oocytes or a sperm necessarily, because it's enough, for example, with a sample of blood, and also provides information about potential transmission to the offspring, which is something useful in case of needing genetic counseling. The problem of gene sequencing is that we need to know which are the genes we need to sequence. But if we talk about fertilization, the main candidate is PLC-zeta. This gene codes for a protein called phospholipase C-zeta-1. This is a protein which is sperm specific. It's the smallest one within the phospholipase family, and it's soluble. We know that this protein can be visualized in the sperm head, for example, in the postacrosomal and the equatorial regions, and it's the main male factor responsible for oocyte activation. Here in this image, you can see a summary of how PLC-zeta works. Once PLC-zeta enters in the oocyte, it binds to its substrate, PIP2, which is present in intracellular vesicles in the oocyte. After hydrolysis by this enzyme, PLC-zeta produces IP3, which binds to IP3 receptors, and this causes a movement of calcium from the endoplasmic reticulum to the cytoplasm. And this increase of calcium will activate the signaling pathway, which will end in meiotic resumption, pronuclei formation, and cell division. There are several studies that support an essential role of PLC-zeta as an oocyte activation factor because this enzyme, this protein, is able to trigger calcium oscillations in both human and mouse oocytes. Also, not so long ago, in 2018, Nozawa and colleagues demonstrated that mouse which were knocked out for PLC-zeta were totally unable to achieve fertilization through ICSI, as well as being unable to generate calcium signaling. And we also know that there are some genetic variants which have been reported that can affect protein function of PLC-zeta and explain fertilization failure. The problem was that before 2018, only 3-4 variants were described. So we didn't know if these variants were common among fertilization failure patients, if they were associated to a particular kind of fertilization failure, or the most important thing, if we could use PLC-zeta gene sequencing as a clinical marker for diagnosis. For this reason, we started a study including a high number of patients with fertilization failure after ICSI. The minimum inseminated number of inseminated oocytes was 4, and they had a fertilization rate below 25%. The total number of men included was 37 patients and 13 controls, and we sequenced PLC-zeta gene sequence in all of them. Here you can see the results. There were 13 patients with fertilization failure that had a genetic variant, at least one genetic variant in PLC zeta, something which represents 35%. Also, in the controls, on the contrary, we didn't find any genetic variant. In total, there were 6 different genetic variants in PLC zeta. 5 of them were missense. 1 of them was frameshift deletion. And the most prevalent mutation was this one, the last one, which is S500L, which affected the C2 domain of the protein and affected 9 different patients. Except for one case, all the patients carried the PLC zeta genetic variant in heterozygosis. Here you can see the 6 genetic variants represented in the secondary structure of the protein. As you can see, they affect different domains. For example, the first one affects the EF-HUNT domain, which is a calcium binding region. The last one affects the C2 domain, which is a domain important for the enzyme to find its substrate. And the rest ones affect the XY regions, as well as the XY linker, which are responsible for the catalytic activity. In the second image, you can see an in silico prediction of how one of these mutations, the frameshift, can alter the structure of the protein. This particular mutation leads to the formation of a truncated protein, which we expect to be non-functional. In a sub-analysis, we divided the patients into groups. One group characterized by abnormal fertilization, and the second group characterized by oocyte activation failure. And interestingly, what we could observe, and in accordance with the role we know PLC zeta has, most of the patients carrying genetic variants were in the group of oocyte activation failure. However, despite this clear association, we needed to prove that PLC zeta variants can indeed cause a problem in the protein and generate the fertilization failure we observe after ICSI. There are different methods to do it, but what we did is to clone the sequence of PLC zeta wild type in a vector, introduce each one of the mutations by directed mutagenesis, produce the mutated cRNA by in vitro transcription, and inject this cRNA inside human oocytes maturated in vitro. And finally, we evaluated if these injected oocytes got activated or not. Here you can see the results. You can see that for the wild type PLC zeta, we obtained an activation rate close to 75%. Also, in the negative control, the sham injection, we obtained 0% activation rate. And what we could observe is that for 3 of the variants, there was a significant decrease in the activation rate, especially significant for the frameshift mutation, in which the activation rate was 0%. There were 2 mutations, 2 variants that obtained an activation rate of, quite high, similar to the one obtained by the wild type. So we consider these variants to be non-pathogenic or benign. And there is the last variant, the S500L, in which we obtained an activation rate close to the wild type, but we do not discard that this variant can really cause a problem in the oocyte activation ability, because similar variants located in the C2 domain, which is not a catalytic domain, it's a regulatory domain, have been reported to be able to cause oscillations when injected in non-physiological levels. So probably what we see when injecting a high amount of protein, mutated cRNA, is that we have calcium oscillations, but probably in a physiologic condition there aren't. Here you can see in this table the clinical data of the patients carrying PLC zeta variants at the moment of being included in the study. As you can see, the fertilization rate was very low, and in many cases there were more than 1 ICSI cycle with fertilization failure. Among the 13 patients, in 10, we applied assisted oocyte activation in the next cycle, and all of them recovered or partially recovered the good fertilization rate we usually observe, and 7 of them achieved live birth after 1 or 2 embryo transfers. This confirms that assisted oocyte activation is a technique that's useful for patients carrying PLC-zeta genetic variants, and it's a good approach to overcome this problem. However, this study I mentioned was published around 2.5 years ago, and what we observed is that not only for PLC-zeta but also for other genes, there has been an increase in the number of studies published associating novel genetic variants with fertilization failure. So we wondered, where are we now? If we know anything more about PLC-zeta genetic variants, and if there are more target genes that we can use to diagnose fertilization failure. To answer these questions, what we use is to start a systematic search within the PubMed database in order to identify all the potential studies which report genetic variants which are associated with zygotic arrest or fertilization failure after ICSI. As you can see, there were more than 100 studies that were retrieved, although only 38 met the inclusion criteria, which were at least 4 mature oocytes inseminated, less than 33% fertilization rate after ICSI, and the use of morphologically normal gametes. For the reason we excluded, for example, globosospermia, which is a severe form of teratosospermia. And after adding some additional studies, the final number of studies included was 46. These studies referred to both male and the male genetic alterations in both the oocyte and the sperm, but I will only talk about the male side today. Here you can see the results. Regarding the male part, we had 16 studies associating genetic variants in different genes with fertilization failure after ICSI. And as expected, the main gene the gene with most variants reported was PLC-zeta1, specifically 28 genetic variants found in 46 patients. In these studies, functional assays were performed in 22 out of 28 variants, assays which were either experimental or in silico, and the pathogenicity of these variants was demonstrated for 17 variants. The approaches to prove the effect of one specific variant are diverse. For example, in the left, you can see an approach which is to measure the activity of phospholipase in vitro. Also, other authors, what they do is to try to measure if a particular variant can affect the localization or the amount of protein within the sperm. Independently of the approach, we can say that there are many variants in PLC-zeta that they affect the wall length of the protein, and indeed almost all of them, 27 PLC-zeta variants, directly affect at least one domain in the protein. In a further characterization, we can say that most of the genetic variants detected for PLC-zeta are missense, and although there are more than 20% variants which are frameshift and nonsense. And what is a little bit surprising is that while more than a half of the patients had both alleles affected, so meaning that most of the half of the patients carried the variants in homozygosis or compound heterozygosis, there is more than 40% of the patients that carry a single variant in heterozygosis. This opens a question, which is if PLC-zeta genetic variants in heterozygosis can cause fertilization failure. This needs further research. It's something we cannot answer right now, but there are some facts that would support this hypothesis. For example, we know that the sperm are genetically different, but they may be phenotypically similar in terms of protein composition, because we know that the spermatids share cytoplasmic bridges interchanging RNA transcripts. Also, we know that the presence of mutated PLC-zeta may affect the ability of wild-type PLC-zeta to trigger oocyte activation. And also the simple presence of a mutated form of PLC zeta could reduce the amount of functional PLC zeta or alter the localization of the wild type PLC zeta. Also, with this analysis, we can try to answer a question, which is which is the frequency of PLC zeta 1 genetic variant fertilization failure. To do it, we analyzed the studies. We analyzed the studies that included multiple patients with fertilization failure. We saw that the percentage of men with carrying a genetic variant in PLC zeta was more or less constant among the studies. It was always between 25 and 35%. So although to answer completely this question, we should need a population study including hundreds or even thousands of patients, we can say that as an approach, 1/3 of the male-related fertilization failure after ICSI may be caused by genetic alterations in the gene of PLC zeta. Also, in this systematic search, we identified some other genes, for example, WPP2NL, which codes for PAWP. 4 variants in 6 patients with fertilization failure were identified. However, we don't really think this is a good marker for the diagnosis of fertilization failure, because despite some authors propose that PAWP can interact with proteins containing WW domain and also activate PLC gamma, this non-canonical pathway to activate the oocyte has not been demonstrated. In addition, it has not been demonstrated that PAWP can cause calcium oscillations in the oocyte. PAWP null mice do not show fertilization problems, and also PAWP does not rescue PLC zeta lack of function when there is a mutation in PLC zeta. On the contrary, the systematic review identified a recent study which found 3 different mutations in homozygosis in 3 infertile men which had fertilization failure after ICSI, characterized by oocyte activation failure. These patients, despite they had an increased proportion of tapered head sperm, they had normal morphology sperm. But in a closer look, the authors realized that these mutations were indeed affecting the abnormal structure of the acrosome, as well as the structure of the perinuclear theca. This is because the mutated actin-like protein 9 cannot interact with another actin-like protein, and the acrosome and the nuclear envelope lose their interaction. This causes that PLC zeta is not localized properly, and the sperm fails to produce calcium oscillation in the oocyte. So to end with, luckily we have some treatment options for patients carrying these mutations. As I mentioned, AOA is useful for patients with PLC zeta mutations. Also for patients, it seems that it will be useful for patients carrying mutations in actin-like protein 9. There are some authors that propose that injection of PLC zeta 1 cRNA would be a good alternative to activate the oocyte in a more physiological way. But this has not been tested in patients and needs further research. And to end with the presentation, I will just give the message that for any infertility problem, especially fertilization failure, we always need to consider both gametes. It is true that in the oocyte, there have been described, there are several mutations described in different genes that can be associated with fertilization failure. But I hope I convinced you that there is an important role of the male also, of the sperm part. And to conclude, as I mentioned, there are different genetic variants which are associated with male-related fertilization failure after ICSI. That PLC zeta 1 is the gene with more pathogenic variants described. And we believe that gene sequencing of this gene could be an option for the diagnosis of patients experiencing failed ICSI outcomes. And of course, future research is required to increase the number of known genetic variants associated with fertilization failure, as well as to identify if there are additional genes which can be useful for this purpose. So thank you very much for your attention.
Thank you, Mark. And now the floor is open to questions from the audience. Do you have any questions for Mark? Well, it seems that the audience are shy, so I'll start. I didn't quite understand if this phenotype, if this failure to to fertilize is something that's specific to ICSI, or are these men? Would these men have also difficulties conceiving through natural means?
Yeah, all these variants were detected in a context of assisted reproduction. So obviously we see that there is fertilization failure after ICSI, but before, all these patients obviously had problems when trying to conceive in vivo. That's why probably that's why they attended to our fertility clinic to try to become parents. So yeah, PLCC, the genetic variants can affect the ability to fertilize both in vivo and in vitro.
Mm. So I guess then that the assumption is that these mutations are de novo mutations in this that the patients carry in this gene.
Yeah, we don't really know the origin. It's probable, it's something possible, because if not, the father could be infertile also. Although we don't really know, because for some mutations there exists the possibility that generate not a complete infertility but also subfertility. Especially in heterozygosis, maybe some patients retain a partial ability to activate. So I don't.
Have you checked if these mutations are present in the general population?
Well, in the general population, we had some controls which are fertile, in which we have seen that there aren't mutations. But in the general population, we should need a large population study, screening hundreds of patients or men, which we don't know.
Well, you have many public databases where you can check SNP allele sequences.
We checked, and most of these mutations are very, very rare.
Okay, so we do have questions from the audience, and so Britta asks, thanks you, and asks, when do you think we should check the PLC Z sequence? After one failed fertilization, after two, only when it is unexpected?
Yeah, it's a good question, because sometimes we wait for 2, 3, well, in many clinics they wait for 2, 3 failed cycles to start doing something. I will suggest that as early as possible, we should sequence, we should try to offer the patients a diagnostic. Perhaps, especially if we don't see any other factor. For example, if we have oocyte donation, we know the donor has fertility proven, and the patient is normosospermic with any apparent problem, with only one cycle of fertilization failure characterized by oocyte activation failure, I should start sequencing the gene.
We have one question now from Marco Freire, who wonders whether the genes that interact with PLCZ could have a similar effect. Have you checked for the network around PLCZ?
Yeah, we looked for potential interactors. However, the PLCZ is somehow simple in its mechanism because it doesn't really need the interaction with another protein in terms that it's released in the oocyte and it literally it goes and finds the substrate. The example I mentioned about ACE acting like protein 9, it's not directly an interaction of PLCZ, but it's somehow related because the mutation of this protein can alter the localization of PLCZ. It's somehow more structural than a direct interaction with the enzyme of PLCZ.
Yeah, and Sandra Lara has quite a pointed question. Is that actually when you treat these patients and you actually make them have children, the baby boys in the sons of these men will also have the same mutations. At least they might, if well, depends if they are homozygous, heterozygotes. But anyway, you are transmitting. You are allowing for something to be transmitted that will probably result in fertilization failures in the next generation. So, is this expected to happen?
Yeah, definite. If it can happen, of course, if we have a mutation which is related to infertility, which is the case of PLCZ, this can be transmitted into the offspring if we find a way to overcome the problem, for example, by using KOA. And in this case, it should be important to offer some genetic counseling to the patients, because the genetic counseling is usually limited to diseases which are somehow more important than infertility maybe, but I think it's something important to know for the patients that probably their offspring will somehow have infertility.
Okay, so and these also you have also answered then Rita's questions about that. So do you think that this would warrant the sex selection in these cases, since the effect then will be much greater on sons than on daughters? Would be
well, in this case, we don't expect. So a girl carrying a PLCZ mutation would not be infertile. In any case, we are not allowed by law in Spain, at least, to select for to do PGT, for example, to select embryos based on a mutation which is infertility related. PGT can also be done only for mutations that are grave, which and appear and don't have a cure in the offspring.
Okay. More questions from the audience? Do we have any other questions from the audience? Going one. Oh. Yeah. And of course, Rita adds that it's not that would not be legal in Spain.
Yeah.
More questions? Then, if there are no further questions, let me thank Marc again for his presentation.